The overall goal of this procedure is to grow primary rat neurons in such a way that their neurites are suitable for visualization by cryo electron microscopy. This is accomplished by first preparing dishes with EM grids for plating primary neurons. The second step is to prepare and plate the primary neurons on EM grids. Next, the vitrification of neurons on EM grids is performed. The final step is to collect images by cryo-electron microscopy, followed by post-processing and annotation. Ultimately, cryo-electron microscopy and tomography are used to show the ultrastructure of neurites in a frozen hydrated state.
The main advantage of this technique over existing methods is that it can be used for 3D visualization of frozen hydrated neurites at nanometer resolution without the use of chemical fixatives, therefore facilitating the assessment of morphological characteristics that are closer to the native state.
Begin this procedure by examining the integrity of holy carbon on the gold EM grids using a light microscope at a magnification of at least 25 times. Make sure the carbon holes are larger than 98% intact. For plating primary neurons, use a Bunsen burner to flame sterilize the EM grids and concurrently render them hydrophilic. Immediately transfer the grid with the carbon side up to the center of the glass bottom dish. Place one EM grid per dish and use only pre-sterilized glass bottom dishes.